to present a small study in which we have applied our network-based methods on the neuroblastoma challenge that David just presented. So first, let me start with the motivation behind using our network-based approach. And it is the following. There is now evidence in the literature that biological network, through their topology, their structure, encodes biological relevant information. And for instance, in a yeast protein network, here the proteins that have more partners in the network also have a higher probability to be essential for yeast survival. And this has been proven a while ago and mostly studied in gene and protein networks. And our idea is to investigate a similar hypothesis, but in patient network. And so this is our strategy. So we are starting from the omics data that were just introduced from neuroblastoma patients. We are actually having two transcriptomic data set for 598 patients. And after filtering, we have in addition genomic data for 120 patients. In addition to the omics data, as already mentioned, we have clinical data. And what we did is to first extract relevant endpoint that we wanted to predict. And so those are the disease risk, disease progression, and death from diseases. Those are binary features that are available for all patients. And so the first thing we wanted to perform on the omics data is dimension reduction. So we selected the most predictive features for each omics dataset and for each of the clinical endpoints of interest. And as a kind of control, we also selected the most varying features, so this time regardless of the clinical endpoint of interest. At this stage, the features that are selected can very well be used to train and test a classification model directly. But what we propose instead is to use those features to derive a patient similarity network in which nodes represent patients and the similarity between their molecular profile is encoded by the weight of the edges. So the thickness of the edges here on this toy example. And so we're using two different methods to build those networks. They both rely on computing the correlation between the molecular profiles, but the second one also makes sure that the network we obtain has a scale-free property. In the next step, we use those networks. We have built one per omic dataset we use, and we actually compute metrics on those networks for all nodes. And so far we've focused on centrality metrics, those 6 that use different definition of centrality to measure how central each node is with respect to its neighborhood or with respect to the entire networks. And so what we propose is to use those network features to also train and test a classification model. Now, for classification task, we define a setup that is very similar to the one described by Zheng and colleagues in the genome biology paper that described the original data. So we use the same train and test samples. We use similar algorithms, and we measure the performance using the accuracy, balance accuracy, and Matthew correlation coefficient. So just to explain and be very clear, the classification task is to use half of the data plus half of the clinical data to train a model, and then use the other half of the data to make prediction about the other half of the clinical data. So with this setup, the first thing we wanted to investigate is whether the network features that we have just defined can be used to build efficient classification models, and also to check whether those models, how do they compare to models that are built by using directly the omics features instead. So the result are the following. What you're looking at is the balance accuracy here for the 3 endpoints of interest, and for the network-based methods in blue, and the omics, what I call the omics-based, in yellow. So what these pictures tells us is that on average for all the clinical endpoints, the performance of both methods are within the same range. And so although you might see small variation here and here, it might look like the network-based methods is giving better results, and the other way around here. Actually, when you look at the numbers, the effect size is really moderate. As can be observed in this table, I just extracted one model for progression, and now you can see not only the balance accuracy, but also the accuracy and the matriculation coefficient. And you can see that the differences are consistent between the two models using the 3 different index, but it's about 1 or 2 percentage points. So in conclusion, we believe that using these network-derived features gives models that are as powerful as the model that are built using the omics features directly. In a second step, we wanted to tackle and address the question of data integration, because what I presented so far was only on the transcriptomic data, so for which we have almost 500 patients. So we wanted to see whether integrating genomic data in addition would lead to better results. So now you're also looking at the balance accuracy, but now you have the network-based methods on top and the omics at the bottom. And now each single data point represents one model that is built using only one of the data set. And so you can first observe that the performance for the genomic data is a lot worse than for the microarray and RNA-seq data. And the second observation is also that both transcriptomic data sets give the similar performances, which is in agreement with the original finding of Zheng and colleagues in their paper. So now the interesting part, when we integrate more data, we see actually no improvement whatsoever in classification performance for this task. It's even worse here. We see a little bit a decrease in performance on average, although we have some models for which we are still able to make sense of the genomic data. So to conclude this part, for this task and in our hands, it looks like the genomic data doesn't really help. Then the third point I wanted to highlight is we've defined 6 different centrality metrics, and we wanted to check whether they were all informative. And so again, you're looking at the balance accuracy, and now each point represents a model that is built using only one metric at the time. And so you can see there are huge variation in performance depending on the metric you're using. And what's still very nice is that this time integrating multiple metrics still give better performance than using the best metric, which is even easier to see when you split the data per endpoints. So in blue you have using all metrics, and in orange here you have using only one of the metric. So you can see that it's clearly superior performance when you're using all at once. So we believe that because the different metrics we have used, they rely on different definition of centralities, they are able to capture different signals that are complementary for this task. And now the last point I wanted to highlight in this presentation, in order to validate here the ability of those features that we have derived to be predictive, we wanted to use an external tool, and therefore we have used ISD, which is a tool that can perform topological data analysis. And ISD was fed again either with the omics features or with the network features. And in each case it will produce a network. And this is the result. So those are also patient network, but each node now can correspond to a group of patients, not a single patient. And edges represent patients that are shared between the nodes. In addition to creating the structure of the networks, we can also add color to the nodes. And here we have decided to color the nodes according to the disease risk of the patients within the nodes. So you can see that in both networks, we are able to actually cluster the nodes according to their disease risk, because this network on the left is built using the omics data, and this one using the network's data. So to conclude this part, we believe that we obtain a similar capacity to cluster the patients when we use the two different set of features. So to conclude, using the neuroblastoma data from the CAMDA challenge, we have observed that the patient network, just like the gene and protein networks, also encode through their structure relevant information, and this time clinically relevant information, because we can use it to predict the outcome of the patients. On average, the performance, I would say, is on a par with the performance of models using the omics data directly. However, the number of features is much less. And for perspective, there are numerous, but I wanted to highlight the two that we are going to do in the following months. I want to include more network features and not only metrics that define the centrality of the nodes to get more information from the network. And also I want to have a look at better integration strategies to try to make more sense of all the data that we have. And so with that, I would like to thank the people involved in the project. I would like to thank Peter Nazarov, Tony Kaoma, Arno Muller, and Songyun Kim for helping me in this project. Also Francisco and Jagat, the two PIs of the project. Let me also thank the Luxembourg National Research Fund for funding, ISD for giving us access to their platform, and of course the CAMDA organizers for making this possible and having me here. Thank you for your attention.
Thank you for your talk. I had a question. If I understand correctly, you create the network based on the omics data, the patient network, and then derive features about what the centrality of the network to use for prediction. I have two questions about that. One is, how is the centrality associated to the response? Is that does more central mean better response? And can you give an intuition of what that would mean? Why would centrality, for example, be associated with response here?
So first, you have to get that centrality. The centrality score we get is not correlated to the similarity of the underlying profiles. So let's say nodes that have similar molecular profile might not have always similar centralities. And to answer the first question, also, we in general higher centrality in this specific case means higher risk or progression. But this is not always the case. In a different disease, in a different context, it's the other way around. In breast cancer, you would have a different, completely inverse observation. So this is not a general rule. It has to be specific for one data set. And what was the second question again?
The intuition of why certain centrality would associate to response.
So in this case, I'm not quite sure, but for other diseases we've studied, it's more like when the majority of the patients, when we're able to, let's say, to cluster patients in group, they might still have similar centrality. Some of the centrality values will be the same. You know what I mean? That you will, even when you have two clusters, still on average one will have higher centrality than the others. And it's even more the case when you have more than two clusters.
Okay, thanks.
Hi, thank you for the talk. So here you're taking sort of a lot of the molecular data that you have and building up these centrality features, but it strikes me that you will lose some interpretability as to what's going on by sort of having these summarizations. Can you talk a little bit about how we would interpret the centrality features and what sort of the meaning of these is? I think this is sort of building on the previous question.
Yeah. So this is something we have not currently looked into, but we have ideas that when we will have more features, we will still have to go back to the original features. So one idea is that when we will have more network features, we'll be able to more easily cluster patients, and then we will in the end have to go back to the omics data that are more representative of those group of patients, and for 1 second forget about the centrality metrics that allowed us to make those clusters. So we'll have to go back to the omics data anyway, because like I said, in different cases, centralities may mean different things. So you cannot hope that you have one explanation that is general enough.
Thank you.
So you've shown us that you have similar performance if you use network-based features and omics-based features. Have you tried combining them together and then selecting the most predictive one, and do you get a better performance if you do that?
So yeah, that's something I need to do. I started to do this, but I don't have the results yet. So I still need to because now what I have is like very few centralities, and even when you do dimension reduction for the omics, you still have a lot more data. So I would still need to take that into account. So far, what I did is use the random forest among the algorithm I presented, because it's one that might still work when you have this large imbalance in number of features. But I still need to do it by do even more dimension reduction to select only a few omics features. Then I will have a balanced data set, I guess.
And obviously you can think of also using deep neural networks, which would do sort of feature selection for free, basically.
Yeah, that would also be nice, yeah. Thank you.
Just a quick question. When you build your networks, do you build them separately for your training and testing data, or using the whole data? So
I'm using the whole data, because otherwise I have not the same scale in centrality. So everything is done, the network building is done for all nodes, okay. But dimension reduction is done only based on training.
Okay.
So it's just that the network needs to be complete. I don't see here train and test. I don't see the test as an external validation of the methods. You see what I mean? So I still want the training sample and test sample to be within the same network.
Okay. Yeah.
Okay. Are there any more questions? Then we will continue.